Hi, I'm Lauren Lee.
And I'm Taylor Fisher, and we are in Period 4 Biotech.
For our experiment, we will be feeding wild-type C. elegans bacteria, which have the dsRNA, which is double-stranded RNA, matching the A gene that we got, which will trigger the RNAi system in the C. elegans.
Using RNAi, we will shut off the gene in C. elegans, and doing so will help us determine the characteristics of the affected gene. While looking at the starter plate for the wild-type worms, you can see that there are many different worms at various life stages, including egg, L1, L2, L3, L4, and adult. In majority, the worms we observed in the starter plate were hermaphrodites. We did not find a single male that we know of. We chunked the wild-type C. elegans onto the feeder plate that had the bacteria with the dsRNA in order to trigger the RNAi in the C. elegans to turn off the A gene within the worms.
In order to chunk, we used a spatula to take out a section of the agar in the starter plate and moved it to the feeder plate. After 6 days, we checked back on the feeder plate to identify any changes on the worm's behavior or morphology.
Upon close examination, we found that our worms, after 6 days of being exposed to the dsRNA bacteria, have begun exhibiting changes in their movements. They now have a twitching motion in their torsos and seem to be doing this action involuntarily. We can hypothesize that the new behavior of the worms is due to an alteration in a gene named UNC22.
The turning off of UNC22 results in distinct twitching motions in the body, which cannot be found in the behaviors of the wild types.
Changes in the worms occur because UNC22 codes for the protein called twitchin, which is needed to regulate muscle contraction or morphology. RNAi, which made the UNC22 gene be shut off, occurs when the dsRNA triggers the RNAi pathway. Dicer cuts up any dsRNA it finds into siRNA, which are small interfering RNAs.
The siRNAs bind to several proteins, forming RNA-induced silencing complex, or RISC. The RISC binds to the matching mRNA and cleaves it. Therefore, the mRNA cannot be translated into a protein, so the gene is silenced. I'm Lauren.
And I'm Taylor.
And thank you for watching our video.